Wishing all the listeners of the Immunology for Everyone podcast by SKA welcome greetings. I am sure the previous episode stirred some thoughts and brewed some ideas to further probe into the specifics of the two APCs. Moving ahead into the concepts of immunology and moving closer to applying it to therapy, SKA is here today with another component of the immune system that contributes significantly to our body's defense action. Take notes, send in your queries, and keep this light inside you thriving. Do share with your peers and fellows. Up next is your host, SKA.
Hello, everyone, and welcome back. Ladies and gentlemen, how many of you have heard about penicillin sensitivity? You all must be knowing that penicillin is very effective antibiotic used for some bacterial infections, but unfortunately cannot be used in some individuals because they are sensitive to this. So this is the reason you must have seen that doctors will, and they should actually, check sensitivity to penicillin by a skin test on every individual before starting this treatment, or they should ask for the history of any penicillin reactivity because it can induce a very severe allergic response, which is called anaphylaxis in many individuals. So, which can be actually life-threatening if it is not managed immediately. So, what is this and how it happens? Physiologically, penicillin spontaneously degrades to reactive intermediates that act as heptins and covalently bind to the self proteins, which can elicit an immune response. About approximately 95% of penicillin degrades to penicilloyl moiety, which is called the major antigenic component. So what are heptins? Heptins are actually small chemical groups that cannot elicit immune response in form of antibody production on their own because they cannot fulfill some of the basic conditions required for the activation of a special type of cell, which are called the B cells. However, when these heptins are coupled to a carrier protein, they become immunogenic because the protein will carry multiple heptin groups that can now cross-link the receptors on the surface of these B cells, leading to their activation. So in addition, some of the antigens which require T cell help, they are called the T cell dependent responses, are possible only because T cells can be primed to peptides derived from the carrier proteins. So in clinical setting, the coupling of a heptin to a protein is responsible for severe allergic responses shown by many people to the antibiotic penicillin, which reacts with host proteins to form a coupled heptin. So that can stimulate an antibody response and cause the reaction. So today we plan to discuss the activation of these B cells. These B cells, they form the primary antibody secreting cells, and they generate the second arm of immune response, which is known as the humoral immune response. We have already talked about this activation of T cells, which makes the cellular immune response, which is the first arm of the immune response, and the second arm of the immune response is humoral immune response, which is very important to have a overall immune response for clearance of a invaded pathogen. So in this process, most of the activated cells, like we talked about the T cell activation, these activated T cells, they differentiate to acquire specialized effector functions, while a smaller portion of activated T cells, they become memory T cells. The effector T cells generally die after performing their function of elimination of the pathogen by direct killing or via activation of other cells like B cells, NK cells, macrophages. So all of them, they are acting against the invaded pathogen. While the memory cell normally become dormant cells and are long-lived cells carrying the antigen specificity. And these memory cells can get activated only when the individual gets exposed to same or similar pathogen in future. So as I mentioned earlier, that the B cells, they are the ones which make antibodies. So just for the knowledge of those listeners who may not have the biology background, an antibody is a protein which is produced by B cell when it detects a harmful substance, which is called the antigen, which may include the microorganisms like bacteria, fungi, parasites, viruses, or some chemicals like toxins, toxins produced by certain bacteria, like you must have heard about tetanus toxoid. So these B cells, they have the capacity to make antibodies against all that. And these antibodies, they carry the specificity for that antigen, which induced their production and specifically binds to the same antigen in order to eliminate the antigen. So antibodies, these antibodies, they are proteins. They are once produced, they keep circulating in the blood for a few weeks to months, and they provide protection against the same pathogen. Besides their role in protection, these also help in diagnosis by detection of that infecting agent. Thereby, they facilitate the start of specific treatment to that infection. So B cells, which are involved in the humoral immunity of the adaptive immune response, as they differentiate into antibody-forming cells, they are known as the plasma cells. Besides this function, we also know that the B cells also are considered as professional antigen-presenting cells, because these B cells, they carry a B cell receptor, which is essentially, and this surface immunoglobulin has the capacity to bind to the antigens on the surface of bacteria or viruses, and that can start the process. So the B lymphocytes, they are also distinct from T lymphocytes in the manner that the B lymphocytes, they are generated in the bone marrow, as the T cells are also generated in the bone marrow, but T cells have to go to thymus to get mature. So that's why they are called T cells. Whereas the B cells, they remain in bone marrow and they mature there and become mature B cells with their B cell receptor on their surface in the bone marrow itself. So that's why they are named as the B cells from the bone marrow. Now let us see how the B cells can get activated. It is a general rule in adaptive immunity that naive antigen-specific lymphocytes are difficult to activate by antigen alone. As we have seen in case of T cells, that they require the antigen to be presented on the surface of antigen-presenting cell when the antigen is bound to MHC molecules. And they also require co-stimulatory signal, which we call the second signal, from the antigen-presenting cells. In case of B cells, there is some kind of similar phenomena, because B cells also require accessory signals that can come either from a helper T cell or in some cases directly from the microbial constituents. So the first signal for B cell activation is delivered through the antigen receptor, which is the B cell receptor on their surface, which is essentially immunoglobulin molecule. And the second signal for most of the antigens is delivered by a helper T cell. So these antigens which require a T cell help for the B cell to get activated, all these antigens are categorized as thymus dependent antigens. So while there are some antigens which do not require T cell help, and these are called thymus independent antigens. So we will talk about thymus independent antigens, which in which the second signal can be delivered by antigen itself or non-thymus derived accessory cells. We'll discuss about them in the next episode. But today we will talk about the T-dependent B cell activation. So as I said, the T-dependent responses require the activation of B cells with the help of a T cell, which is called T helper cell. But the condition is that this T cell must also recognize the same antigen, which means that before the B cell can be induced to make antibody to an infecting pathogen, a T cell specific for the antigens from this pathogen must first be activated to produce the appropriate activated helper T cell, because this helper T cell has to help the B cell for activation. So, and we know that the T cells, they get activated by antigen presenting cells. Generally, dendritic cells activate the naive T cells. So, although we also know that the T cells basically recognize short peptides because the antigen is first processed inside the dendritic cells, and only smaller peptides are loaded onto MHC molecule, which are displayed on the surface of these T cells, and then they are recognized by the T cells. But B cells, they can even recognize globular kind of proteins or soluble proteins or some protein patterns on the surface of the pathogen. So the epitope recognized by T cell may not be the same as the epitope recognized by the B cell, but the condition is that these two epitopes, the T cell epitope and the B cell epitope, should be linked. They should be present on the same antigen, which is a bigger kind of antigen pathogen. So that is the crucial point, that they have to be the T cell, the peptide which is recognized by the T cell be a physical part of the antigen recognized by the B cell. So let's move forward. So to fulfill this condition for the activation of B cell by the T cell, having primed with the same antigen depends on the availability of antigen specific B cell to concentrate the appropriate peptide on its surface along with MHC molecules, because that's how the T cell will recognize. For this job, the B cell that binds to a particular antigen, they are about 10,000 times more efficient at displaying the peptide fragments of that antigen on their MHC class 2 molecules than the B cells that do not bind to the antigen. So the sensitized T cell will help only those B cells whose receptors have bound to an antigen containing the peptide that the T cells can recognize. So this particular intricate mechanism of coupled antigen epitopes is known as linked recognition. So this is a very important phenomena, which is very important for T-dependent activation of B cells. The phenomena of linked recognition was originally discovered through studies on the production of antibodies to haptens. So I told you that haptens are very small chemical groups, so they don't basically elicit the antibody responses on their own because they cannot recruit T cell help, because they are very simple small molecules. However, when hapten is coupled to a carrier protein, they become immunogenic because the protein will carry the multiple hapten groups on their surface that can now cross-link the B cell receptors, which is very important for B cell activation, the cross-linking of B cell receptors. In addition, the T cell dependent responses are possible because T cells can be primed to the peptides which are driven by the carrier protein. So once a hapten is conjugated to carrier protein, then the immune response is generated. So T cells as well as B cells, they get activated and you get antibodies generated against the hapten. Which can later on react with the hapten very nicely. So I told you about penicillin, sensitivity, which is this kind of phenomena because of this. So one is the requirement for linked recognition has important consequences for the regulation and manipulation of the humoral immune response. And one of that is the linked recognition also helps in self-tolerance, because as the T cell epitopes and the B cell epitopes are physically linked, they are present on the same antigen. So the T cells, when they are presented by an autoantigen, the self-antigen, the autoreactive T cells, I told you, they become tolerant because the antigen presenting cell, when it acquires a self-antigen, will not upregulate the co-stimulatory molecule expression, which is important for T cell activation. Because the antigen presenting cell have to see a pathogen associated molecular pattern to get activated and express the co-stimulatory molecules. So in case of self-antigens, that co-stimulatory molecules are not expressed, and the antigen presenting cell sends a tolerizing signal to the T cells. And in a similar manner, B cells also get tolerized. So there is a self-tolerance generated, which is very important. And another important application of linked recognition is in the design of vaccines. And I will give you a very interesting example of a vaccine which is against the Haemophilus influenzae type B. And you have heard about Hib vaccine, which is given to your children. Hib is a bacteria which can cause illness that can lead to potentially deadly brain infection in children. This may also cause diseases such as meningitis because of the inflammation of the meninges, which is a covering of brain and spinal cord. It can also cause bloodstream infections, pneumonia, arthritis, and infections of other parts of the body. Now, the protective immunity to this pathogen is mediated by antibodies, which need to be against the capsular, polysaccharide. This bacteria is covered with a coat, which is a polysaccharide coat, and the antibodies have to bind to those. In adults, they make very effective immune response to these polysaccharide antigens via other mechanisms, which I talked about, the thymus-independent mechanisms. But such responses are very weak in infants because their immune systems are immature. So below 5 years, the immune system is not mature enough to produce the B cells, which can make antibodies by thymus-independent pathway. So to make an effective vaccine for use in children below 5 years, the polysaccharide of this particular bacteria is linked chemically to a toxoid, which is called tetanus toxoid. And we know that tetanus toxoid is a very good immunogen, and a successful vaccine is already available. So when this polysaccharide is linked to a tetanus toxoid and that is given as a vaccine, then the B cells, they bind to the polysaccharide component of the vaccine, and they can get activated by the helper T cells, which are specific for the peptides of the linked toxoid. So both T cells and B cells, they get activated for a physically conjugate of tetanus toxoid and the polysaccharide. So a very successful vaccine which protects children against Hib infection. So in this context, how the B cells and T cells, they have to interact. It's a puzzling, feature, because the antibody response is how an antigen-specific B cell manages to encounter a helper T cell with an appropriate specificity to the same antigen. That is the condition. So this question arises because the frequency of naive lymphocytes, which are specific for any given antigen, is estimated to be between 1 in 10,000 or maybe 1 in 100,000. So 1 in a million case, you will find a specific antigen carrying specific receptor. So the chance of an encounter between a T cell and the B cell that recognize the same antigen is calculated to between 1 in 10 to power 8 or 1 in 10 to power 12, which is very low, very less chance. And achieving such an encounter is far more difficult challenge than getting the effector T cell, which is activated by its interaction with the antigen presenting cell. Because in that case, only T cell has the T cell receptor, which is antigen receptor, whereas antigen presenting cell is just presenting the antigen. So even in that case, where the chances are much, much higher, still this happens. So in case of naive T cell activation, where does the answer comes from? The answer comes from that antigen specific T cells, they get trapped in the lymph node where the antigen presenting cell is there, which is displaying the antigen. So they get trapped because we know that when an antigen is introduced into the animal, it is captured and processed by the professional antigen presenting cell, which move to the lymphoid organs, the lymph nodes, and the T cells, which are coming from thymus, they basically pass by those antigen presenting cells, which are displaying the antigen, and they continuously keep flowing till a rare T cell, which is carrying the specific receptor, will bind to that dendritic cell and get trapped. This trapping clearly involves the specific antigen receptor on T cell, but also stabilized by activation of other molecules, which are called the adherent molecules and chemokines, because they all bind to the T cell. And somewhat similar, very ingenious experiments using mice which are transgenic for rearranged immunoglobulin genes, they have shown that in the presence of appropriate antigen, the B cell with antigen-specific receptor are also trapped in the T cell zones of lymphoid tissues by a similar mechanism. So it is a antigen specificity which helps in trapping of that B cell into those areas. And so an encountering antigen, these migrating antigen binding B cells are arrested by the activation of adherent molecules and engagement with the chemokines. Such chemokine receptor like CCR7 is also upregulated. So the trapping of antigen-specific B cell in the T cell zones provides an elegant solution to the problem, how the T and B cell specific for same antigen, they come together for active interaction. So this is a very beautiful phenomena, very interesting. And then interaction with pre-sensitized helper T cell activates the B cell, leading to their clonal expansion. I told you that T cell get activated, they clonally expand. So that antigen, that T cell having the antigen receptor, clonal expansion. Similarly, B cells also, once they get the activation signal, there is a clonal expansion with the formation of a primary focus. So here at the border of T cell zones and the B cell zone, because they live in separate zones, but at the border they meet, both types of lymphocytes will proliferate and for several days, to constitute the first phase of primary humoral immune response. So what is the take-home message today? What I have discussed is the B cell activation by thymus-dependent antigens. They require both binding of antigen by the B cell receptor and interaction of the B cell with the antigen-specific helper T cells. The helper T cells recognize the peptide fragments derived from the antigen, which are internalized by the B cell and displayed on the B cell bound to the MHC molecules. Then the initial interaction occurs in the T cell area of secondary lymphoid tissue, where both the antigen-specific T helper cells and antigen-specific B cells are trapped as a consequence of binding the same antigen. And this is known as linked recognition. So this interaction that leads to clonal expansion of antigen-specific B cells and formation of primary focus of antibody-forming plasma cells. So guys, I would like to close here today, and we'll take up the very interesting aspect of further differentiation of these plasma cells which have come from the activated B cell to make very high affinity antibody. And that is again a very interesting phenomena which we will discuss next time. So keep listening and also read some of the interesting articles which I have referred here. Till then, goodbye and thank you.